Hello everyone, IDDI here. For those of you who are already connected, welcome. We're just waiting for a few more to connect. We can see that they're downloading, so please bear with us for a few more minutes, maybe 2 to 3 minutes maximum. Thank you. I hope you're still with us. I'm still waiting for a few more people to connect. It won't be long now. Thank you. So I think, yep, that's it. So let's start. So hello everyone, my name is Beverly Baker and I'm account coordinator with IDDI. I'd like to welcome you to our first webinar on statistical validation of biomarkers, and I must say we're particularly happy and proud to say that we have over 270 registrations today, so hence the couple of minutes delay. I wanted to give you a quick overview of IDDI for those of you who don't know us that well. Since 1991 we provide specialized services in phase 1 to 4 clinical trials. And these include trial design, biomarker validation, data management, statistical analysis, report writing and supporting contacts with the FDA and EMEA. We have developed our own web systems, an interactive web response system for patient randomization and treatment allocation, and also internet coding of medical terms. These web systems are integrated with Marvin, a professional EDC system. Over 20 years, IDDI has developed a thorough experience in oncology, cardiology, ophthalmology, and biosimilars. So, the content of today's webinar will focus on statistical validation of biomarkers, and this presentation will take about 45 minutes, and this will be followed by a question and answer session based on the questions some of you have asked us at the time of your registration. You'll understand that because of the larger number of attendees, it would be rather unreasonable to try to answer further questions online, but you will have the opportunity to ask them later as soon as you exit the webinar. Today our speakers are Dr. Marc Beuze, and he's founder and chairman of IDDI based in Louvain-la-Neuve, and associate professor of biostatistics at the Hasselt University in Belgium, and also Professor Thomas Berzikowski, who's Director of Research at IDDI and Research Professor of Biostatistics, Bioinformatics at the Center for Statistics of the Hasselt University. So I think what I'll do now is pass you over to the experts, and I would just like to say, enjoy the webinar.
Hello everyone, my name is Marc Beuze. I'm going to begin the webinar by setting the scene, and then afterwards Thomas Berzikowski and I will take turns to cover the different aspects that we would like to cover as indicated on this slide. I will first define what we mean by biomarkers and the types of biomarkers we'll be discussing. Thomas will then discuss prognostic biomarkers. I will turn to predictive biomarkers. Thomas will handle surrogates. I will discuss trial designs, and then Thomas will conclude. So this is the outline of our talks. And let me begin with a few definitions. This definition of a biomarker was proposed by a biomarkers working party, and if you're interested in more details, you can refer to the paper indicated at the bottom of your screen, a paper that appeared recently in Nature Reviews in Clinical Oncology. And a biomarker was defined by this working party as any characteristic that can be objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacological response to a therapeutic intervention. Now, a biomarker can be many different things. It can be anything you can measure in a patient that is not a clinical endpoint. And these include physiological biomarkers, image-based biomarkers, and increasingly recently interest has focused on cellular, molecular, genetic, and genomic biomarkers. Now, we will in fact discuss in turn prognostic biomarkers, predictive biomarkers, and surrogate biomarkers. A prognostic biomarker is one that forecasts the course of disease regardless of treatment. A predictive biomarker is one that forecasts the likely response to a specific treatment. A clinical endpoint is something that measures how the patient feels, functions, or survives. And typical endpoints that we will discuss will come from our examples in oncology, and they will include survival time or distant metastasis survival time and other endpoints of that nature. A surrogate endpoint is a measurement that provides early and accurate prediction of both a clinical endpoint, such as survival, but also the effects of treatment on this endpoint. And finally, what do we mean by validation? In this talk, validation will mean the confirmation by statistical methods that a candidate prognostic, predictive, or surrogate biomarker fulfills a set of conditions that are necessary and sufficient for its use in the clinic. So we will use validation in this specific meaning. It is convenient to classify biomarkers by how many times we measure them. And there are really two classes of biomarkers. The first class is biomarkers that are measured once before treatment starts. And the second class concerns biomarkers that can be repeatedly measured over time. That means before, during, and also possibly after treatment. The first class, the biomarkers that are measured before treatment, can be themselves divided into prognostic and predictive biomarkers. And we will spend most of the time today discussing those two classes of biomarkers. The biomarkers that can be measured repeatedly over time include pharmacodynamic biomarkers that are commonly used in early clinical trials, and we will not discuss those at all. But we will turn our attention towards the end of the webinar to surrogate biomarkers or surrogate endpoints that can be used in lieu of our clinical endpoints of interest. Let's now turn to prognostic biomarkers.
Thanks, Mark. Hello to everybody. Yes, let us focus now on prognostic biomarkers. As Mark indicated, prognostic biomarkers are biomarkers which can predict the likely course of the disease for individual patients, and they can be used for several purposes. One of them is patient stratification in clinical trials. This is perhaps relatively trivial. It allows us to gain in efficiency when we organize a trial. Or they can be used for treatment decisions, and this is perhaps more interesting because we can, for instance, decide that patients who are likely to have a bad prognosis might need to receive an aggressive treatment, whereas for those who are likely to have a good prognosis, we may actually resign from the treatment or delay the treatment until symptoms appear, which will then trigger the need for a treatment. Let us first describe a little bit the situation for the prognostic biomarkers. So we'll consider the situation where we have a binary biomarker which can be either present or absent for individual patient. In these two figures, on the x-axis we have the two treatment groups, let's say standard and experimental. And on the y-axis we have the value of a clinical endpoint with the larger values indicated a better value or better prognosis for the patient. So this picture shows a situation where actually nothing, the left picture shows the situations where actually not much is going on. There's no treatment effect because the mean value for the standard and experimental group is equal, and actually this is also this is the case for marker present and marker absent group. Moreover, there's no difference between the two groups of the groups defined by the presence of the marker. So in fact, there's a situation of no treatment effect and no difference between the no difference in the marker groups at all. On the right hand, we see a slightly different situation. Again, we don't see a treatment effect because the mean values for the standard and experimental group are exactly the same. However, we see the difference now between the two groups defined by the presence of the markers, which means that in this case, patients who have for whom the marker is present have a better prognosis than patients who do not have the marker. And this means that by knowing the marker status of the patient, we could on average say whether the patient would fare better or not. So this is how we tend to think about the prognostic biomarker, and this also leads to the idea of how we could validate a biomarker, which basically means that I'm sorry, I have a problem with coming back. Okay, so the thing would be that what we in our validation of the prognostic biomarkers, what we should look at is we should look at the association measures. We might look at association measures to see whether there's association on average between the biomarker and the clinical endpoint. This is relatively straightforward, and we can do this without a special design for a study. We can do this retrospectively. We can do this in a prospective study in a clinical trial. But the main question is whether this type of analysis, or whether there's enough information or enough association between the biomarker and a clinical endpoint to predict the course of disease for individual patients. Now, this is an important point, and I will try to discuss it in a little bit more detail, starting first with this theoretical representation. So what we see here on this plot is the plot of the false positive fraction and true positive fraction for a binary biomarker, which is used to predict binary status, good, bad prognosis status of the patient. Now, these curves, as you can see, there are different curves, and they correspond to the increasing value of the odds ratio, which measures the association between the biomarker and the clinical endpoint. So, if we focus first on this first line, which is basically the line corresponding to the odds ratio of 1, of no association between marker and prognosis, or in this case about the good or bad prognosis, we can see that actually there's no situation in which we could get high value of the true prediction, true positive fraction with small false positive fraction. Basically, if we are here with, let's say, true positive fraction of 80%, the false positive fraction is 80%, so what shows that we have we make an error in terms of claiming too many false positive results. On the other hand, if we want to have small false positive fraction, we have also very small true positive fraction, so we make errors in terms of the claiming too few patients as true positive. Now, if we increase the association between the marker and the endpoint, or the, let's say, the prognosis, we can see that we start to at least have some situations where the true positive fraction can be large and false positive fraction can be small. But this requires us to have relatively high value of the odds ratio. For instance, if we take odds ratio about 16, then we can see that we can have a true positive fraction of about 80% with a false positive fraction of about 20%, which would be acceptable. However, this this value of 16 for the odds ratio is relatively extreme, and it's probably rarely seen. And if you want to see even better results, ever better classification or discrimination results, you would have to go to the values of odds ratio of like 36 or more. Mind you also that even if the odds ratio is about 16, we can still have a situation, for instance, where the true positive fraction is about 90%, but the false positive fraction is 40%. So this still means that we may be doing fine in claiming in one way, so claiming true positive status, but we may be making a wrong error in terms of making too many false positive calls. And this theoretical consideration, which shows that association between the measure of association between two endpoints, between marker and the clinical endpoint, does not capture the discriminative power for a marker, is illustrated by an example. And this example will be presented on the next slide. Yes. So this is the example of the gene signature which was presented by Van der Wever in the New England Journal of Medicine. So in the figure on the left-hand side, you see the two survival curves which show the probability of remaining metastasis-free as a function of time. And these two survival curves are for good signature and poor signature patients. So based on the signature which was constructed from expression levels of 70 genes, the authors of the paper divided the patients into two groups, and you can see that there's a marked difference in the survival, metastasis-free survival for these two groups. And this is actually markedly different from the situation, from the curves which are constructed for the low-risk and high-risk patients in Saint-Gallen based on the classical Saint-Gallen criteria, which are just clinical factors based. This is also shown here with this p-value, which is highly significant, which shows that indeed, well, the difference which we see between these two survival curves is statistically significant. So this would show that on average there's a strong association between the signature value for a patient and the prognosis in terms of the metastasis-free survival. Now, let us focus a little bit more on the results, on the more concrete results which were published in the paper. So here we see a cross-classification of patients by the prognosis, by the signature status, so poor or good prognosis, and we have also classification with respect to the true disease status, meaning whether the patient had this metastasis within 5 years and whether they were metastasis-free during the 5 years. So this is the cross-classification table. Yes? Yes?
Thomas, I'm sorry to interrupt you. We receive a lot of complaints that your voice doesn't come through very clearly. So I don't know if your microphone can be adjusted, but some people have difficulties hearing you. Could you speak a little further away from your mic?
I'm sorry, this was too close to the mic, yes?
This is better.
Yes, I'm sorry about that. I'm very sorry.
Fine.
Okay, so as I said, we look at this cross-classification table, and we can calculate the odds ratio based on that. And based on this these data, we can see that the value of the odds ratio is about 15, which suggests a strong association between the marker status and the disease status. However, if we look at the properties of this, let's say, signature as a diagnostic test, we can compute sensitivity or true positive fraction as equal to about 90%, which is very good. However, if you look at the specificity, it is only about 60%. So in terms of a diagnostic test, it means that this test wouldn't be too specific in predicting the, say, prognosis or predicting the disease status or disease-free status of the patients. Similarly, if we look at the positive predictive value and negative predictive value for the marker, we get negative predictive value, which is about 90%, which is again good value, but the positive predictive value will be only 63%, basically meaning that we again cannot, well, given the positive result of the test, it's only 63% probability that the patient indeed will have bad prognosis. So this means that this high association, which we saw based on the odds ratio, was actually not telling us everything about the predictive accuracy of this marker. And if we come back to this picture which I showed you before, we can also see this clearly from the curve which I presented for the odds ratio of 16. For the gene signature we were having the odds ratio of 15, so it's more or less this curve which I just circled. So if we remember, our sensitivity was 91%, our odds ratio was 15, and if we look at the corresponding specificity, it has to be about 60%. So this means that indeed this is an example which shows that even if we have relatively strong association measured by the odds ratio, in terms of the discriminative power of the marker, of the gene signature in this case, we are not doing too good job in terms of the specificity of this signature. This can be explained further by another example, which I've taken from the paper by Patrick Royston and colleagues in Journal of National Cancer Institute. This example is based on a clinical factors, not on the biomarkers, but it is illustrative to explain what happens in our previous example. So what Patrick Royston and colleagues did, they took, this was the example in advanced renal cancer, they took a couple of factors including performance status, hemoglobin level, white cell count, and logarithm of time from diagnosis of metastasis to randomization, and they constructed a prognostic index based on a model for survival of the patients. Then they divided patients in 3 groups according to the level of this prognostic index, and they plotted survival curves. And you can see these curves here, and there's a clear and marked differences. There are clear and marked differences between these 3 groups, indicating that indeed the prognostic index based on these clinical factors on average very clearly distinguishes patients with a good, moderate, or poor prognosis. However, if you look at the individual values of survival for patients, this picture emerges. So here we have survival time in months on the log scale, and here we have our 3 groups created on the based on the prognostic index. And here we have well, let's say like counts of patients who have particular value for overall survival. The circles are patients who were censored and their values were imputed based on the model. So what you can see is that, well, clearly on average the survival times for the poor prognosis patients are shorter than for moderate or good prognosis patients. But there's a considerable overlap between all these 3 distributions, which basically means that indeed on average the prognostic index is associated with the survival time for these patients. But for individual prediction, we have still too little power to really distinguish good prognosis and bad prognosis patients because of this considerable overlap. And this is exactly the problem, that if we validate a prognostic biomarker based on association measure, we look on the association which tells us how much the differences in the distribution of the clinical endpoint between the different groups which are based on the biomarker, but it doesn't tell us the whole story about the possible discrimination of about the power, discriminative power of the biomarker for individual patients, and this should actually be taken into account when we look at the validation of a prognostic biomarker.
Okay, let's move on now to predictive biomarkers. The uses of predictive biomarkers, just as for prognostic biomarkers, can be a stratification of patients in trials, but predictive biomarkers can also be used to select patients for trials, as we shall see, but also, and more importantly, to make treatment decisions. So we'll detail that in a moment. But coming back to the slide Thomas showed before to explain what a prognostic biomarker was, it's the same slide here, but to illustrate what a predictive biomarker is. So now we have again the clinical endpoint on the vertical axis and the 2 treatment groups, the standard and the experimental, on the horizontal axis. As we see here, a predictive biomarker is one which predicts the effect of the experimental treatment compared to the standard. In this particular instance, we've assumed that there was no benefit to patients who are marker absent to receive the experimental group, but there is a benefit for patients who indeed are biomarker present or biomarker positive. Now, obviously, a biomarker can be prognostic and predictive at the same time, and so we can look at the whole sort of array here, where we have biomarkers that are neither predictive nor prognostic, biomarkers that are purely prognostic, in other words, their effect is independent of treatment, biomarkers that are purely predictive, that means their effect is only linked to treatment, and then a combination of both, biomarkers that are both prognostic and predictive. I think one of the best examples of a predictive biomarker in oncology has appeared recently in the literature, and this concerns EGFR mutation in patients with non-small cell lung cancer. This paper in particular is very interesting. It shows the results of GEFITINIB, or IRESA, given to patients with non-small cell lung cancer as compared with a conventional chemotherapy regimen consisting of carboplatin and paclitaxel. So here is the main figure of this paper showing the probability of progression-free survival as a function of time for the two groups of patients, those randomized to receive GEFITINIB, which is the curve in green, and those randomized to receive the conventional chemotherapy, the curve in orange. And as we see on this graph, actually, this is a very unusual situation where the curves cross each other at about 6 months. So this curve seems to indicate that chemotherapy does better than GEFITINIB initially, and after 6 months there is a reversal whereby patients receiving GEFITINIB begin to do better. Now this of course is a very unusual situation, and one in which the hazard ratio indicated here of 0.74 doesn't have any meaning anymore, because the hazard ratio overall is in fact a mixture or an average of the hazard ratio over the whole time axis. And that may not be terribly meaningful because, again, GEFITINIB does worse initially and better later. Actually, fortunately, it was discovered as this trial was ongoing that EGFR mutation is an important predictive factor in these patients, such that patients who have the mutation, which is the subset represented here, actually benefit from GEFITINIB. And you see on this graph that the GEFITINIB arm is doing better at all times than the chemotherapy group, whereas patients who are not mutated, patients who have EGFR negative status, in fact are harmed by receiving GEFITINIB instead of receiving the conventional chemotherapy. And so putting it all together, if we now put the four curves on the same graph, we see very clearly that the effect of chemotherapy is independent of EGFR mutation. Patients who are mutated and patients who are not mutated benefit about equally from chemotherapy. However, in contrast, patients who receive GEFITINIB do much better if they are mutated and much worse if they're not mutated. And so here we have a case of a qualitative interaction, that is an interaction whereby the treatment effect is positive in one subset of patients and negative in the other subset. So this would be a qualitative interaction. Now, in order to validate a predictive biomarker, we would need to show such an interaction, and obviously in most common situations, the interaction would be expected to be less than what we've just seen. But this is in fact the principle that we should adhere to, that we would test an interaction, and only if the interaction is significant would we claim that we have a significant predictive marker. We need randomized evidence, obviously, to test for an interaction in order to estimate the treatment effect by the biomarker levels reliably. And the problem with these interaction tests is that the power of these tests is very low. So let us illustrate that the power of these tests is very low. First of all, let's look at the situation of EGFR mutation in patients with non-small cell lung cancer, the example that we've just seen, where patients are treated with IRESA or GEFITINIB on the one hand and chemotherapy on the other hand. The treatment hazard ratio overall, ignoring the EGFR mutation status, is 0.74. The interaction hazard ratio, which is the ratio of the hazard ratio of patients who have the mutation divided by the hazard ratio of patients who do not have the mutation, is 0.48 divided by 2.85, and that's a hazard ratio of 0.17. So we see from the comparison of these two hazard ratios, the treatment hazard ratio and the interaction hazard ratio, that the interaction effect size is much larger in this case than the treatment effect size. And that's why the interaction was actually highly significant in this trial. In reality, we should not expect to have such large interactions or significant interactions. And let us see on this slide by how much we must increase the sample size of a trial in order to detect an interaction that has a certain size as compared to the treatment effect hazard ratio. Suppose first that the hazard ratio of the interaction is about equal to the hazard ratio of the treatment effect. In that case, we see on this slide that we should multiply the sample size of the trial by a factor of about 4 in order to have the same power to detect the interaction as the power that we initially had to detect the treatment effect. So a factor of 4 is already a substantial increase, but it's still doable. If now we are interested in an interaction that is about half the size of the treatment effect, and that might still be clinically worthwhile, then we would have to multiply in order to detect this interaction. We would have to multiply the sample size of the trial by a factor of 16, and this of course becomes almost undoable in most cases. So the bottom line is that unless we have very strong interactions, perhaps even qualitative interactions, as in the example we've discussed, it will be very difficult to power a trial to detect an interaction with any kind of statistical power, reasonable power. So this is the main problem with predictive biomarkers. It's very hard to detect them reliably. And let's now move on to the next topic, which surrogate biomarkers.
Okay, thanks, Mark. Indeed, so this is the third type of biomarkers, which, from the point of view of validation, is probably the most difficult one. Okay, so surrogate biomarkers are biomarkers which we try, as Mark said at the very beginning, which we try to use in lieu of the clinical endpoint of interest. And obviously the main potential use of surrogate biomarkers would be to assess the treatment effect earlier, because we could perhaps get the information about the treatment effect on biomarker-based endpoint earlier than on the ultimate clinical endpoint of interest. Also, it may happen that, for instance, the biomarker-based endpoint may have may for instance be more sensitive to treatment effects than the ultimate clinical endpoint, and this would have implications in terms, for instance, of a sample size which would require for the detection of the treatment effect. So the idea would be to take a surrogate biomarker and use it instead or replace the clinical endpoint with the biomarker in order to speed up the assessment of the treatment effect, or to do this more efficiently. But the question is, when can we do this? And this is the question about the validation requirements for the validation. So at this moment, the only feasible approach which can be used for validation of surrogate biomarkers is based on meta-analytic approach, which basically means that we require data on the biomarker and clinical endpoint from multiple randomized trials which were organized in the past with some treatments for the particular disease. And what we need to show is, first, to show that there's a strong association between the biomarker and the clinical endpoint, because this in a way shows that the biomarker is prognostic and gives us some evidence that there may be biologically meaningful association between these two endpoints, between the biomarker and the clinical endpoint, which justifies its use. What is more important, however, is that we need to show a strong association between treatment effects on the biomarker and the endpoint, because then if we have such a strong association, we can build a prediction model which would allow us to reliably predict the treatment effect on the clinical endpoint from the effect on the biomarker. So this is an example in advanced colorectal cancer which shows how the validation could proceed. In this example, we took 10 historical trials which compared 5-FU plus leucovorin with 5-FU alone, or royalty tracks, and we have built up a prediction model based on these 10 historical trials. And then we looked at 3 more recent trials, which we called validation trials, which compared 5-FU plus leucovorin with 5-FU plus leucovorin plus irinotecan, or there was one trial which compared 5-FU plus leucovorin and irinotecan versus 5-FU plus leucovorin and oxaliplatin. This picture is the main picture in the paper which we published with these results. What you can see in this picture is that on the x-axis there's a treatment effect in terms of the hazard ratio on progression-free survival, and on the y-axis you see the treatment effect in terms of the hazard ratio on overall survival. The blue circles are the treatment effect estimates, the pairs of the treatment effect estimates for the historical trials, for these 10 historical trials. And based on these points, we estimated the linear regression, which is our prediction model based on this historical data. The dashed curves are the prediction limits which are associated with this model. So basically, what you can see is that the historical trials, the estimates of the treatment effects on the progression-free survival and overall survival for these historical trials are pretty tightly associated with this regression line, which allows us to build up a prediction. And basically what it means is that on the value, taking for instance the value of 1.25 as the hazard ratio for progression-free survival, we could predict that the effect on the overall survival would be something about, well, maybe 1.2 or something. So there's a little bit of attenuation. And what you can see that, for instance, that this prediction would be very good for the trials which compared 5-FU plus leucovorin and combined with irinotecan. So these are the two trials indicated by the square, and you can see that they almost sit on this prediction line, which indicates that in their case we could actually take the effect on the progression-free survival and predict almost exactly the effect on the overall survival which was observed in these trials. For the oxaliplatin trial, the situation is slightly different. It is a little bit off from the prediction line, but it stays nevertheless within the prediction lines, and importantly it's also indicated the prediction lines exclude the line of 1, so they would indicate that also statistically significant effect on the survival would be expected. So if we can construct such a prediction model, and if the prediction coming from this model are reliable in the sense that they can be validated to, for instance, to give a meaningful and let's say credible prediction, this would be a signal that we could use a particular biomarker as a surrogate for a clinical endpoint. So, as I said, this is a methodology this is actually the only feasible methodology at this moment, and there are examples which I list here where this methodology was actually applied. So as you can see, these are examples in oncology. These are examples of potential surrogate endpoints for survival in oncology. One comment here would be that actually in all these examples the potential candidate for the surrogate was endpoint like progression-free survival or disease-free survival. Only in advanced prostate cancer example, something of a nature of a biomarker, namely the PSA antigen, was used as a potential candidate for a surrogate. So there are not many examples of using biomarkers as potential candidates for surrogates for survival, at least in this case in oncology. Nevertheless, as you can see, there's a range of diseases where this exercise for validating a candidate surrogate were performed. This example by Dan Sargent, together with the example which I just showed you, were taken by the FDA and formed the basis for the decision by the FDA to allow progression-free survival or disease-free survival as a candidate surrogate, as a surrogate for overall survival in colorectal cancer trials. So basically what I could sum up is that the validation of surrogate biomarkers requires individual patient data from multiple trials, and knowing how difficult it is to get such a data and analyze them, this indicates that the validation of surrogate biomarkers is a complex issue. One important point is that we need to have the association which we see in historical trials may not necessarily hold for a new treatment which may have a different mechanism of action. So one of the problems with the use and validation of surrogate biomarkers is whether the prediction model which we build up on the historical data can be extrapolated to a new treatment which may have a different mechanism of action, and therefore which may perhaps deviate from in terms of the treatment effects from what we have seen in the previous trials. This means that perhaps the pure statistical validation in this case may not be enough. Maybe it has to be combined with some biological evidence which would support this possibility of extrapolating. as I said, this is a complex issue, and there are many issues related to the validation of the surrogate endpoints, and there are many methods, and therefore it's impossible to present it in this webinar. But in a book which I and Mark co-edited with Gert Molenbergs, there's a broad description of many of these issues and methods, where more details can be found.
Thank you, Thomas. I would like to now turn to trial designs, and this could easily be the topic of a whole webinar. So in the next few minutes, what I'd like to do is just to glance over some of the designs that have been proposed, and perhaps at a later stage we could have a whole seminar or webinar on this issue. In fact, if you think of it, if we just wanted to validate a biomarker, we could do this without a prospective trial. However, if we need to look at the clinical value or the clinical utility of the biomarker, then we must have some randomized evidence. How can we, for example, validate the clinical utility of a prognostic biomarker? This is a possible design. In fact, we would need to compare in a randomized fashion a biomarker-based strategy with a conventional or standard or usual strategy based only on clinical and pathological factors. So on this slide you see that patients can be randomized between a biomarker-based strategy, and then they're assigned to two different treatments, standard or experimental, depending on their risk as assessed by the biomarker, versus another group of patients who are randomly assigned to a clinical assessment of their risk, and again are assigned to standard treatment or experimental depending on their clinical risk. And so if the biomarker is indeed important and predicts the outcome of the patients better than the clinical factors, we would expect this group, where the biomarker served as the decision-making tool, to perform better on average than the group in which the biomarker information was not used. So this is a possible design. I'm not saying it's a perfect design. In fact, it has many problems, but it's certainly a possible design. Probably a better design is to actually use the information from both the biomarker and the clinical risk, and to combine these two pieces of information. So in this design, the patients would be assessed for their clinical risk factors first, and then they will be assessed also for the biomarker. And so patients could be actually low risk by both the biomarker and the clinical risk factors, in which case they would receive standard therapy, or they could be high risk by both the clinical factors and the biomarker, in which case they should all receive some more aggressive, perhaps new experimental treatment. And it's only for those patients in whom the biomarker assessment and the clinical assessment differ that we would actually randomize these patients between the standard therapy and the new experimental therapy. And this randomization is justified because the patients are classified low risk by one system and high risk by the other, or vice versa. So in a sense, the clinicians are not sure that these patients need to receive standard or experimental treatment. This idea was actually implemented recently in a trial called the MINDAC trial, performed by the EORTC in Europe. And this trial will randomize 6,000 patients with early breast cancer, and will perform the gene signature that Thomas already described in the webinar, and will actually assess the risk group of these patients by the 70 gene signature. It is expected that about 60% of the patients will be high risk, both by the clinical factors and by the signature. Those patients will all receive chemotherapy. At the other end of the scale, 10% of the patients, it is assumed, will be assessed low risk both by clinical factors and by the signature. And these patients will not receive chemotherapy. They may receive endocrine therapy if appropriate, but they will not receive chemotherapy. And then the 30% remaining patients, who are the patients of interest here, are those patients in whom the clinical pathological factors and the 70 gene signature risks are discordant. So these patients will then be randomized between receiving chemo or not receiving chemo, and in fact this is equivalent to making the decision based on either the clinical pathological risks or on the gene signature risks. And so patients will be randomly assigned to having a treatment based on their clinical pathological risk or based on their gene signature. So half of them will be randomized in each of these groups to chemo and the other half to no chemo. We could do this a little differently, and this is the option that has been chosen in another trial that I will describe shortly, whereby the biomarker, instead of classifying the patients between a low-risk group and a high-risk group, as was the case for the 70 gene signature, actually now classifies the patients between three groups: a low-risk group, a high-risk group, and then an intermediate risk group. And this is in fact how Oncotype DX works, and I will show this in the next slide. The point of this design is to randomize the patients, again, for whom the clinicians are substantially uncertain. And those are the patients with intermediate genomic risk. These patients will be randomized between standard and experimental. And the patients with low risk, just as in the MINDAC design, will receive standard, which is no chemotherapy, and the patients with high risk will receive the standard for their risk, which is to receive chemotherapy. So here is the design of the TAILOR-X trial. It's again a trial for early breast cancer patients. 10,000 patients will be randomized, and in fact all of these patients will undergo the Oncotype DX scoring system based on a piece of their tumor. If the risk group, as assessed by Oncotype DX, is high risk, patients will receive chemotherapy. If the risk is low risk by genomic risk, they will not receive chemotherapy, and patients who are in the intermediate risk, which patients who have an intermediate score by Oncotype DX, will actually be randomized between chemo and no chemo. And so this will prove or disprove that the Oncotype DX has clinical utility. Now we could think of other designs than the designs just covered so far. Another design that would be appropriate for a prognostic factor would be a biomarker-based strategy compared to a non-biomarker-based strategy in which all patients receive the same standard treatment. So in this trial, if indeed the biomarker provided information on patients who are high risk and therefore need to receive a more aggressive treatment, we would expect the biomarker-based strategy group to fare better on average than the patients who receive standard treatment without the biomarker assessment. The problem, of course, of this design is that if the experimental group had an effect in all patients, regardless of their biomarker status, and that means including the low-risk patients, then patients who receive the experimental group would do better, and therefore this group, the biomarker-based strategy group, would tend to fare better than the standard treatment group, regardless of the value of the biomarker. And so this design is not satisfactory from that viewpoint, and it was replaced, or another design was proposed in order to replace this design, where patients are now randomized in the non-biomarker-based strategy group. Patients are now randomized between the same two treatments as the biomarker-based strategy group, and the proportion of patients who receive standard and experimental is in fact equal to the incidence of the low-risk and high-risk group by the biomarker. So in this case, we avoid the problem that if the experimental treatment is better than the standard, we would not expect to see a difference between these two groups, except if the biomarker had in fact some utility. Now, of course, the difference we expect to see between these randomized groups here is very small, and therefore we are talking about really large trials here to validate the clinical utility of this biomarker. What about predictive biomarkers? As I said before, in order to prove that a biomarker is indeed predictive for the effect of a treatment, in this case the effect of experimental compared to standard, we need actually to have a biomarker negative group and a biomarker positive group. And so we could stratify patients in a prospective design. We could stratify patients by the biomarker status and randomize the patients in those two strata between standard group or experimental, between standard treatment and experimental. And if the biomarker is truly predictive, we would expect the benefit of experimental treatment to differ in the biomarker negative group and in the biomarker positive group. So the test of the null hypothesis here would be that we would test if these two effects were the same, and if we rejected that null hypothesis, then we would claim that the biomarker is indeed predictive. Now sometimes there is a lot of prior data that seems to indicate that the biomarker is predictive, and in that case it may not even be ethical to randomize patients who are biomarker negative. I'm thinking, for example, about the KRAS mutation in colorectal cancer patients, Now, in such situations where it's well known that biomarker negative patients cannot respond to a certain experimental treatment, it's unethical to give them the experimental treatment. And so here we would be talking about a selection design whereby patients who are biomarker negative would all receive the standard treatment, and only patients who are biomarker positive would be randomized between the standard and the experimental. This is why this design is called the selection design. And the only question of interest in this design would be to look at the effective treatment among biomarker positive patients, regardless of how the treatment would fare in biomarker negative patients. Now, like I said, these designs have been proposed. Some of them are actually currently used, but I think we would need much more time to discuss the pros and cons of these designs, and this might be an issue for later.
Okay, thanks, Mark. So I guess this is time to conclude. Obviously, there's, as we mentioned with Mark, there are a lot of issues, and certainly some extra webinars would be needed to cover them all. So let us conclude about the biomarker validation. As I presented, validation of prognostic biomarkers is relatively straightforward. However, I think that in all exercises aimed at validation of prognostic biomarkers, more focus on discriminative properties of biomarkers is needed, because the association-based validation does not capture this aspect of prognostic biomarkers to a large extent. As Mark said, validation of predictive biomarkers is more complicated, and mainly due to the limited power of the interaction test. And this is the main issue in this for this type of biomarkers. For surrogate biomarkers, perhaps the validation is the most difficult issue due to the required data, meaning that we require meta-analytic data and more importantly individual patient meta-analytic data, and also because the assumption about the possible extrapolation of the results of the historical trials to a new treatment may be questionable. If we come back to validation of biomarker utility based on the designs which Mark has just explained, the current designs, the feature of these current designs is that they require large sample sizes, mainly due to the fact that the randomization is confined in this design to a biomarker-based subgroup, which very often is of low prevalence, and therefore the sample size for the trial has to be inflated. Also, the sample size has to be inflated because in some of these designs, as Mark alluded to, there's a treatment dilution, so the difference of the treatment effect will be small, and therefore the sample size has to be increased. So clearly, in terms of validating of biomarker utility designs, we need some novel designs, and certainly this is something where the research is going on.
Well, thank you both for that. Okay. Thank you, Thomas. Thank you, Mark. I think we have a bit of time for some questions and answers now. We received 4 questions before the webinar. One of the questions is, somebody would like to be interested to know if you had any comments about the validation process for safety biomarker, i.e. validating a biomarker of drug-induced injury.
Well, perhaps I'll take this one. So I see two major issues here. Well, the answer is yes, we could try to do this, but I would see this as a more complex question than even for the treatment effect, for finding a surrogate endpoint for a treatment effect, mainly because of 2 reasons. One reason is that, well, safety signals can be of very low incidence, and actually it may be difficult to find to validate a biomarker or for such a low incidence signal. We've done some simulations in a setting where indeed we were looking at the binary endpoint, clinical endpoint, which would be of low incidence, and where we were thinking about using a continuous biomarker measures as a potential surrogate. And it was very difficult, and we found that huge sample sizes would be, and huge number of trials, of previous trials would be required for the validation in such a setting. So this may be one of the problems. Another problem which I can link to this assumption that you can, let's say, extrapolate from the previous experience to the new treatment is that if you have new treatment of a new mechanism, the safety may not necessarily correspond to whatever you saw in the past. So while perhaps in the terms of the treatment effect you might expect that the extrapolation might work, in terms of the safety this may be even more difficult. So I would say that this assumption that you may use previous data to say something about the validity of a biomarker for safety of a new treatment, this assumption may be even more, say, problematic, and that would be one of the additional or tough issues to solve with the validation of a biomarker for a safety endpoint.
Okay, thanks. Second question is, could you validate a novice biomarker against an existing, well-accepted biomarker? Who wants to answer that one?
Thomas, you want to give it a try?
Okay, I'll give it a try. Well, my thinking is as follows: well, again, one point would be that you could think of one issue, and this means that let's assume that you have established indeed an association between a biomarker and a clinical endpoint, and then because you don't want, it's easier to get the data on biomarkers, you want to validate a biomarker, a new biomarker, just for the established biomarker, not using the clinical endpoint data. Now the problem which may be there is that you risk the fact that there will be additional uncertainty about the association between the novice biomarker and the clinical endpoint because it's judged indirectly through the established biomarker. So there I would say that of course one could try to do this, but there I would see the major problem or major issue in the fact that additional uncertainty would be introduced there by doing this in an indirect way. So like saying, okay, I have a validated biomarker, now I validate a novice one against the established biomarker, and therefore I can only say something about the association between the novice biomarker and the clinical endpoint only indirectly through this established biomarker, and this means that there will be extra uncertainty about how useful the novice biomarker is. So this is one thing which comes to my mind with respect to this question.
Okay.
Yeah, I'd like to add to this perhaps in general terms. I fully agree with you, Thomas, that one of the problems when we look at validation or so-called validation studies is that very often people are happy if they see a correlation between a supposed biomarker and a clinical endpoint, and they tend to forget the uncertainty and the random variation. And of course, as statisticians, this is the one thing we worry about, so that we don't need to look just at the association between two quantities, but we need to look at the predictive value of this association. And this implies not just the prediction, but the limits around the prediction as well. And that, I think, is a key message from this webinar.
The third question is, do we require prognostic biomarkers to have better predictive ability than commonly used prognostication systems, such as adjuvant online for breast cancer or the Framingham risk score for cardiovascular disease?
Yeah, I think I can answer that one, and I can perhaps answer it using the example of the 70 gene signature that Thomas described before. When we validated this 70 gene signature using a data set of data from independent sites, and we tried to reproduce the results that had been found in Amsterdam, we noticed indeed that the gene signature had some prognostic impact on the outcome of interest, which was the distant metastasis-free survival. And we were a bit disappointed that the odds ratio, or the hazard ratio, if you will, of the gene signature was not very large. However, when we looked at clinical features or clinical factors in the same population of patients, we also noticed that the best prognostic factors that are currently used by oncologists who treat breast cancer didn't do any better than the signature, such that if we used, for example, the Nottingham Prognostic Index or the adjuvant online system, the hazard ratio using these systems was no better than the hazard ratio of the signature. And so we may be wanting too much of a biomarker as compared to the clinical factors that are in use commonly for most tumor types. And that is, I think, something we need to keep in mind. We want the biomarkers to be truly prognostic or truly predictive, but we shouldn't put the bar too high as compared to the factors that the clinicians already use today based on simple clinical factors or pathological factors.
Okay. Very last question is, can we use an incompletely validated surrogate endpoint in the design of a registration phase 3 trial and take the uncertainty about the surrogate into account in the design of the trial?
Mark?
Okay, yes, I can take this one as well, at least partly. I think you can definitely do this and try to validate a biomarker as you go doing your randomized phase 3 trial for registration of a new drug. Of course, you will not be able to convince the agencies in using the data from a single trial that you both have a valid surrogate and that you also can predict the effect of your treatment on the true endpoint if you don't have a significant effect of treatment on the true endpoint in that very trial. However, I think validation of a biomarker in a randomized trial is always useful, and it's supportive information. If, for example, as we had the case recently, you have a trial in which a biomarker was highly significantly in favor of a treatment, but the clinical endpoint failed to be significant, probably because of the small size of the trial, and this was in an orphan indication, then I think you begin to have arguments to tell the agency that the effect of treatment was really, absolutely without doubt on the biomarker. It failed to reach significance on the clinical endpoint because that's a much higher hurdle to reach. But I think it's supportive information that can help in a registration file, and we've used it recently, at least in one instance, with some success at the EMA.
I can add to this. So Mark covered this part regarding the question regarding the use in the registration trial. What I would like to add to that, something which is perhaps linked to what Mark has already said, is that indeed, if you would like to use a surrogate in a trial, one of the points is that if the surrogate is used, it does come with some uncertainty around it. So this, as I described in these slides on the validation of a surrogate endpoint, there is this prediction model which would serve for translating the treatment effects from the biomarker scale to the treatment effects on the clinical endpoint. Now, this is a model which has uncertainties related to that, and these uncertainties should be taken into account when you design the trial based on the surrogate, or when you design a trial using the surrogate endpoint or biomarker. And well, this can be done, and one could think that, well, you can quantify the extra uncertainty which comes from the use of a biomarker, but then this uncertainty should somehow be taken into account, for instance, when sample size is calculated or power calculations are done, and this is something that certainly should be considered where like more formal use of a surrogate biomarker is conceived for a particular trial.
Okay, well, thank you very much. I think that's all we have time for in terms of questions and answers. As I said before, you'll have the opportunity, as soon as you close the webinar down, to leave further questions on the survey, which we would appreciate you filling in, and Mark and Thomas will compile them and send the answers to these questions together with a copy of today's presentation very soon. Now, as you can see on this slide here, this is just the first of a series of webinars, and the next one is scheduled on September the 3rd, so please pencil this in your agenda. We'll send you a formal invitation together with the login information very soon also. So that's all that's left for me to say, and I just wanted to thank you again for having attended, and goodbye for now. Bye.
Thank you. Bye-bye.